Hi, hello friends, myself A.Kiran, year 2019-55, and my topic in Path 372, diseases of horticulture crops under management, is mango anthracnose. So, today let us discuss briefly about the mango anthracnose. So, the word anthracnose means coal. So, the spots produced on leaf, that is dark spots. So the disease is often referred as anthracnose, and the anthracnose in mango invades stem, leaves, inflorescence, and also even fruits. So the anthracnose is a worldwide most important disease that causes huge loss to the mango production and farmers, and mainly in the areas where high humidity prevails. So it is the field phase and post harvest phase. We can say that post harvest phase is directly linked to the field phase, where initial infection starts, in where the disease starts from wings or leaves, later spread to flowers causing blossom blight, and then later to the fruit causing drop of fruit set or formation of mummified fruits. So when we talk about symptoms, in leaves we can observe irregular or circular shaped black necrotic spots on both surfaces of leaves. Later these spots coalesce and form large necrotic areas. So frequently you can see in leaf margins, and such severely affected leaves usually curl. Again, lesions develop primarily on the young tissues, and conidia are formed, and these lesions observed in all of the ages. In older leaves, lesions do not develop, but latent infections or dormant conditions we can see until the tissue senesces. So growth then resumes, and fruiting structures are produced in necrotic tissue. Under favorable conditions, conidia are dispersed and invade the young wings, causing wing dieback in some cases. So when we go on through inflorescence of panicle, the anthracnose symptoms be like the panicle anthracnose or blossom blight can affect both inflorescence, stalk, or an individual flower. So infection reduces the fruit set and production considerably. In the stalk, elongated dark grey to black lesions appear, and the blighted flowers are dry. Their color varies from brown to black. Small fruits can be infected and aborted. The large fruits are aborted because of their physiological causes, prophetically invaded by Luteotrichum gliosporites, on which they sporulate profusely. Take after harvesting, post-harvest anthracnose, there is an appearance of brown to black lesions on the surface of fruit with indefinite border. After initial establishment on the fruit, the fungus remains as dormant or in latent phase as a pinpoint infections until the fruit begin to ripen. Then dark depressed circular lesions will develop on the fruit, and they become enlarged and increases rapidly in their size. Then lesions of different size coalesce and they form and cover the extensive areas of the fruit, typically in a tear stream pattern developing from basal towards the distal portion of the fruit. In severe cases, the fungus even penetrates into the fruit pulp. In advanced stages, the fungus produces acervuli, an abundant orange to small salmon pink masses of conidia. When we talk about disease cycle, let me tell you in a new way. Okay. When we talk about disease cycle, the primary infection is through acervulus or saprophytic mycelium on conidia in the infected plant debris. The spread is through rain splashed conidia. When a rain with certain intensity falls on the infected parts, the conidia may get spread due to the rain splash to the nearby side. When they fall onto the leaves, causes anthracnose symptoms on leaf foliage. When the rain splashed conidia fall on the panicle, the infection leads to the formation of blighted flowers. And when the invasion occurs in the aborted fruits, there will be a formation of mummified fruits. Wherever they fall, as they fall on leaf, panicle or fruit, they again sporulate and leads to the formation of acervulus and further spread of conidia through rain splash. After initial establishment in the fruit, the fungus remains latent or dormant as pinpoint infections until the fruit begin to ripen. In the quiescent infection, the conidia on the fruit surface produce an mycelial outgrowth, that is aprosorium, into the epidermis of fruit. Then dark depressed circular lesions then develop on the fruit and increase rapidly in size. These lesions have different coalesce and cover extensive areas of fruit, typically in tear-shaped pattern, developing from basal towards the distal end of the fruit. Basically, it is restricted to the peel, but in severe cases, the fungus also penetrates even the fruit pulp also we can see. And in advanced stages, the fungus produces acervuli on the surface and abundant orange to salmon pink masses of conidia appear on the lesions, and later they spread to other mango fruits. We can refer it as a post-harvest anthracnose. So this is the disease cycle of anthracnose in mango. So when we talk about favorable conditions, so optimal temperature for the production of conidia is from 25 to 30 degrees Celsius, and frequent rainfalls, which is a favorable condition, helps in the dissemination and spread of disease causing epidemics, and also the high relative humidity above 90%, above 95%. These are the favorable conditions for the more dissemination spread of the disease. So when we go through management, since the development of disease is mainly based on the relative humidity, so the establishment of orchards in areas which having well defined dry season to allow the fruit development in condition unfavorable for the disease development. Pruning the trees to allow the air flows freely through the tree canopy to reduce the relative humidity. Wider plant spacing will inhibit the occurrence of severe epidemics, and the collection and destruction of diseased twigs fruits is also a good management practice. The mango is harvested at mature green state and stored for 2 to 3 months at 10 to 12 degrees Celsius before ripening. Since mango is harvested at green mature stage and stored for at 10 to 12 degrees Celsius before ripening, since the fruit is sold to the buyers still remaining in the tree, the problem of post-harvest control of anthracnose is passed from grower to exporter. Storage fungi via cause rotting in the mango fruit. Storage fungi via anthracnose causes rot in the fruit and causes the loss in quality of the fruit. So another management is after post-harvest management is hot water treatment of mangoes at 55 to 60 degrees Celsius was found very effective against the anthracnose of mango. And when we take post-harvest anthracnose disease control, disease management, we can use dip in solutions like benomel 500 ppm or chloranthanil. Dip in benomel solution 500 ppm or thiobendazole 1000 ppm for 5 minutes is found to be more effective against the control of these anthracnose disease. Chemical. When we talk about the chemical way to control the anthracnose of mango, we can recommend carbendazim 0.1% or chloranthanil 0.2% at 14 days interval till the harvest. So this is the economic importance, symptoms, disease cycle, favorable conditions and management of anthracnose in mango. With this, I hope you understand well. So last but not least and Everest, thanks from the bottom of my heart to Guru Reddy sir, Professor, Department of Plant Pathology, SV Agricultural College, Tirupati, for assigning this topic. So this is Kiran 55 signing off.